guys, let's review just a little bit from the last lesson. What type of bonds hold the DNA together? So what you should say is there's really two types of bonds. We've got covalent bonds that are between the sugar and the phosphates in the backbone. Okay, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then you have in here the hydrogen bonds that are between the nitrogen bases. So remember, the nitrogen bases are your A's, T's, G's, and C's. And so you have the hydrogen bonds that hold those together. Which bases are purines and which are pyrimidines? Adenine and guanine are purines, and cytosine, uracil, and thymine are pyrimidines. Ag-cut, that's what I remember. And then what happens in transformation? So that's when one cell, in the case we were talking about was bacteria, takes in the DNA from an external source and incorporates it into their genome. So that is a transformation. So let's go ahead and get started today. Today we are talking about DNA replication. So you will have some questions in Google Classroom about DNA replication, so it's imperative that you watch this video. The questions aren't super hard or tricky, but they're just very basic questions about the process of replication to make sure that you're watching and understanding and following along. So most of them you should find right in your notes if you copy down the notes as we go through. So DNA replication is called a semi-conservative replication model. And it took a while for scientists to understand that. But basically what that means is that each new DNA molecule consists of half of the old strand and half of the new strand. So here in picture A, we have our parent molecule, okay? These are our nitrogen bases, A's, T's, G's, and C's, held together by hydrogen bonds. That's what these cutouts here represent. Notice the A, it's one of those 3-ring structures, so it appears a little bit longer there. The T is one of the 2-ring structures, so it appears a little bit shorter there. And it's imperative that the A's and the T's pair together so that it's the right distance all the way across. If that A tried to pair with this G here, that would not work out properly. But once those hydrogen bonds are broken, you have the 2 strands that are separated as you see here, and then the daughter strand is made. This light blue strand is made complementary, that's the term that we use, to the parent strand. And so each new strand of DNA has got one parent and one daughter, okay? And so that is the semi-conservative model. It has one old strand and one new strand. That's what that means, okay? So there were a couple other models that they had tried out. The conservative model, basically where you had one old strand and one new strand, and then a dispersive model where you had a couple of different options of each. But that's not what they found. The way that they did this was they labeled the DNA with different isotopes, and they were able to see which, how it worked out. Okay, so when we talk about DNA replication happens in an area called a replication fork. Okay, and so it's kind of a region where the DNA is unwound. So remember, the DNA is a double helix, and it's held together in the middle by hydrogen bonds. And so if we want to replicate the DNA, we have to open up that strand, and that area that's opened up is referred to as the replication fork. Now, where the replication begins, at the spot on the fork where that begins, is known as the origin of replication. And that has a specific area of nucleotides. Now, remember, in a eukaryotic cell, our DNA is linear, okay? It's a long strand of DNA. And so there can be many replication forks or many replication bubbles on the strand. So replication happens at many different spots. It makes it much more effective. It doesn't just start at the very beginning and go to the very end. there are many spots of replication, many replication forks, and they go until they essentially run into one another to copy the DNA more rapidly, okay? Now, in a prokaryotic cell, prokaryotic cells have circular DNA. It's called a plasmid. We'll talk about those more when we do a little unit in bacteria. It's about 2 units out. But they have a circular DNA called a plasmid. And so unlike eukaryotic DNA where the replication proceeds, you have a couple of different forks that's going at many different times, that's not how you have it in bacteria. You have a single replication fork and it goes both ways until the whole DNA molecule is copied, okay? So a little bit of difference there between prokaryotes and eukaryotes, okay? So in DNA replication, we have several enzymes that are responsible. Now, remember, enzymes are always going to end in ACE. So anytime you see something that ends in ACE, that is going to be an enzyme. Now, there are a few here. I think there's about, 4 or 6 that we're going to talk about today. And you may wish to make like a little note card or a flashcard for them. Sometimes that is helpful for me in this lesson because there are a lot of enzymes to keep track of. But the first one is DNA helicase or helicase, either way you prefer to say it. And it is the zipper, okay? So this replication fork, as we saw just a moment ago, has to open up, okay? Here's our replication fork. It forms this bubble here. And so the way that happens is through an enzyme called DNA helicase, okay? It unzips the double helix at the replication fork, separating the strands so replication can occur, okay? Now, in order to hold that replication fork open, you have something else, and that's called single-stranded binding proteins. And so those proteins bind to the strands, okay, right here, essentially, on each single strand, and hold that replication fork open to keep the DNA open so replication can happen and keeping it from rolling back up on itself and repairing, okay, because if that's not held open, that's what's going to happen, okay. Now, topoisomerase. If you're in class with me, I'll kind of show you how this works, and if y'all remind me, I'll show you the other group tomorrow. But topoisomerase is an enzyme that basically goes in. Once you open this up, I want you to think about this as like 2 strands. If you had 2 strands here and that were, coiled together, and you went in and you pulled these 2 apart. This is hard to visualize, but it's so much easier if I can show you in class. And you pulled them open, okay? What's going to happen is these parts of the strands are going to coil tighter. When you open this section up, the parts on the side are going to get tighter, okay? Because they're going to have to pick up that slack. And so what topoisomerase does is it comes in here and it kind of cuts the strand and it unravels it a little bit in a couple of different places to loosen that strain on the DNA molecule, okay? So if you read here, it says it helps relieve strain on the DNA by breaking, swiveling, and then rejoining the DNA strands, okay? So it goes in, cuts it, unwinds it a little bit, and then puts it back together so that the DNA is not so wound up so tightly there, all right? And then the next one is DNA polymerase. So there are actually lots of polymerases, but we are going to specifically talk about DNA polymerase 1 and DNA polymerase 3. So let's talk about DNA polymerase 3 first. So polymerase 3 adds DNA nucleotides to the complementary strand, okay? So if the original parent strand says T, DNA polymerase is going to bring in a A, okay? If it says G, it's going to bring in a C, and vice versa. That's the job of polymerase, to add to the growing strand, okay? Now, there's several polymerases in eukaryotes. Those are always, I think there's at least 10 or 11. Those are always indicated with Roman numerals, but we only need to know a couple of them. And generally, as far as the AP test is concerned, if you just know it's a polymerase, then you're probably good. You don't have to specify so much between which is which, okay? All right, let's talk about this primer. So in order for DNA polymerase to bind, to be able to start adding nucleotides, you have to have a primer, okay? So the primer is interesting because it's made up of RNA, okay? And it's synthesized by an enzyme. We know it's an enzyme because it's an ACE called primase. So primase, this pink enzyme here, synthesizes this RNA primer, and that primer has to be there before DNA polymerase can bind. And remember, DNA polymerase is like the super important enzyme because it's bringing in all of those new nitrogen bases to essentially make this new DNA strand, okay? Now, the primer is a pretty short piece of DNA here. Now, this is a small, not a full-scale diagram, but it's just about 5 to 10 nucleotides long. And again, that DNA polymerase 3 can't bind if the primer is not present. So the DNA starts at the 3' end of the primer, okay? And it continues to move and adds nucleotides to the 3' end, okay? So this is the 3' end on the old strand in the parent strand. So down here, that's the 5' end on the daughter strand. That means the 3' end on the daughter strand is right here. So the primer is going to move in this direction, okay? 5' to 3', okay? Nucleotides added on new strand. This is so important, okay? These directions are really important. DNA is synthesized on the new strand in the 5' to 3' direction. That means it's reading the parent strand in the 3' to 5' direction, okay? We're making it in the 5' to 3' on the new strand. We are reading that doesn't look like an A, 3' to 5' on the parent strand. That may be one of your questions on your Google assignment today, okay? So DNA elongates on the 3' end, all right? Now, where do these nucleotides come from? Well, we said it was the job of DNA polymerase, right? Specifically DNA polymerase 3, but they come from a nucleoside triphosphate. So that's what you see here, this nucleoside triphosphate. So you notice it's got the base. Remember, a nucleotide is a sugar, which was here, our 5-carbon sugar, deoxyribose, a phosphate group, and a nitrogen base. And so we've got these things here. But what else you have is a triphosphate, a nucleoside triphosphate. And so all we need is a nucleotide. And so we've got to get rid of those extra phosphates. And you see they're released here. They're referred to as pyrophosphates, are released here. And so when you break that bond off of these pyrophosphates, then you release energy. And that energy is used to form the bond here, okay? Remember, when you break a bond, it releases energy. But to build a bond, it requires energy. And so again, what we have here is energy coupling, using the energy from the broken bond here to drive the addition of that bond here, okay? So we see energy coupling. and then that added this nucleotide right here. Okay, so that's where it's coming from. All right, so moving on to DNA replication. DNA polymerase can add nucleotides to the 3' end. That's what we just said of the growing strand. Thus, the new strand is said to grow in the 5' to 3' direction. Okay, so just a little bit of an overview of what we were talked here. We've got the primer synthesized by primase. This is the origin of replication. This is the origin of replication right here on both ends that we reached in there and pulled those pieces apart. All right, you're going to have the single-stranded binding proteins over here that are going to hold that replication fork open. You're going to have the topoisomerase down here to relieve some of the strain on the coiled strands over here. And then you're going to have everything's moving in the 5' to 3' direction. So here's 5', here's 3'. On this strand, this would be 5', and this would be 3'. So notice that both of those strands are being replicated at the same time in opposite directions. Okay, same time at opposite directions. Both started at the origin of replication. Now, something else that you might notice, and we're going to talk about this more in just a second, is that these are long, smooth lines. Okay? Those are referred to as the leading strands. All right? And then back here on the back side, you have these little short lines. All of them, though, beginning with a primer. Okay? And those are referred to as the lagging strand. So let's talk about these. The leading strand is created as the replication fork progresses. You only have to have one primer for each leading strand, and it's synthesized in a long continuous segment, as we just saw in that picture. The lagging strand is moving away from the replication fork. It's got to have a primer for each piece, and they're made discontinually in a series of segments called Okazaki fragments. Now that term is important, Okazaki fragments. You will see that on an AP exam. So each of these represents an Okazaki fragment. So they're happening behind the area of the replication. So remember, replication's happening this way for the top strand, this way for the bottom strand. And so that Okazaki fragments, those lagging strands, are being synthesized behind the replication fork. Okay? And then essentially at the end, they'll all be sealed together to make one nice, smooth DNA strand. We can't just have all these chunks and pieces. Okay? All right. I think that's got us here. This is showing our DNA polymerase that's going on and adding these new nucleotides. Our primer here. We're not going to talk about the sliding clamp right now. And then, yeah, I think that's got everything for here. All right. So the other polymerase that we're going to talk about is DNA polymerase 1. And so what the job of DNA polymerase 1 is to come back in and take out these primer segments and replace them with DNA. Okay, so remember, at each fragment, at each Okazaki fragment, you have a primer section, and then also in the leading strands, you also have a primer section. And so those are RNA, and they have to be taken out and replaced with DNA. And so that's the job of DNA polymerase 1. Okay, and then finally, another enzyme is ligase. Ligase is the glue. And the job of ligase is to seal everything together and make one nice, smooth strand, okay? Joining those covalent bonds in the sugar phosphate backbone, okay? So let's start at this picture from the top. We have our primer, okay? Our primase is the pink enzyme. Our primer is the red piece. It's moving in the 5' to 3' direction. DNA polymerase right here. The sliding clamp just kind of keeps it open and holds on to it. But the DNA polymerase is adding these new nucleotides, again, going in the 5' to 3' direction. This is a short little piece, an Okazaki fragment. You see it's just short right here. And so when that primer runs into the back, when that polymerase runs into the end of a primer, it pops off, okay? When it hits the end of, or the start of the next Okazaki fragment, the polymerase comes off, all right? And then the primer is replaced. I'm trying to see. Yeah, this here is our polymerase, and it's going to replace this primer right here with DNA, okay? And then this here is the ligase. This is the polymerase 1. And this is the ligase, and it's going to seal it all together, okay? Now, it's also going to replace this one too, but that just didn't show that part. It just showed this one step, okay? Going to seal it all together. All right. So here's the whole picture. We have, again, our primer is the red piece. Being added, or where the being synthesized by the primase. The DNA polymerase is bringing in the new nucleotides. This is a lagging strand. This short little segment here is a lagging strand. So up here, notice we have this long, nice blue segment. That's our leading strand. So these are also called the Okazaki fragments. DNA polymerase 1 is going to replace this primer with DNA instead of RNA, and then the DNA ligase is going to seal it all back together. Okay, here's our single-stranded binding proteins right there that's holding it open. Maybe this is topoisomerase. Hard to say what that is. I'm going to guess it's maybe topoisomerase. All right. And we see the replication happening in both directions here. All right. So there's those major proteins that are proteins. Well, they are proteins because all enzymes are proteins. But there's those major enzymes that you were to know today. So if you want to make some flashcards of those, I would highly recommend that. And review this lesson, and then go ahead and head into the Google Classroom and answer those questions in the Google form about replication. Y'all have a great day.